This is the beginning of the second day of our symposium, and we have the supervisor talk 3 with Professor Lilian Schweyer, Pereira Martins, and Professor Lilian is from our department and from our program, and she works with story and of science, and she is always willing to help the program. So every time we ask to Professor Lilian to help us in speakers and to get part in committees, she's always ready to help. So thank you, Lilian, for all your effort in your program, and I'll turn the floor to you. Okay, thank you.
Thank you very much, Professor Milton. First of all, I'd like to thank Professor Milton and also Professor Tomás for the opportunity of talking about the research line, history, philosophy, and teaching in evolution and ecology, some research and its results. Well, this presentation has 2 parts. The first one, I will introduce the line in which we dedicate ourselves, me and I and other professors, that are Professor Motokani and Professor Fernanda Bram. And so, I will present also some students, besides the supervisors. I will present also the students, the proposal of this line, its scope, and as well as some results. And in the second part, I will talk about the research that has been carried out at the laboratory of history and theory of biology. That the laboratory, my laboratory, the students and also researchers. I will start by telling you a bit about the history of this research line, how it was proposed and so on. So, in 2011, Professor Marcelo Motokani, Professor Fernanda and I contact the coordination of the program of Comparative Biology, and it was compounded by Professor Márcia Bitondi at the occasion and Professor Ricardo Macedo. So we talked to Professor Márcia Bitondi. And we told her that we would like to join the program and also to create some research lines. And so she told us that it was necessary to propose one line. And so we decided to propose this line that encompasses several kinds of research. So we proposed at first history, philosophy, and teaching of biology. After that, it changed to history, philosophy, and teaching of comparative biology. And nowadays, it is history, philosophy, and teaching of evolution and ecology. Okay? So, Professor Marcia told us that besides proposing this research line, we should also suggest courses, disciplines for the students, and we had also to have students interested in work with us and develop their research under our guidance. And so, besides other things that were required. So, we proposed the line. We were accepted as supervisors at the program. And nowadays I will talk a bit about the scope of this line. Okay? So this line encompasses historical and philosophical studies focusing on the growth of biological thought, the construction of concepts, theories, models, history and philosophy of biology, and science teaching and learning, epistemology and didactic of biology, biology teaching and learning in different levels and space, all this. Each one of these options has its particular methodology, peculiarities, and approaches. We proposed during this time, since 2011 until today, these disciplines: university, training, teaching, and production of knowledge. That is disciplina PAI in Portuguese. Topics on history, epistemology, and science teaching. Topics on history of zoology, introduction to the didactic of biology, biological diversity, and philosophy of biology. So all these disciplines belong to this research lab. Well, now I will present some results, some research that arises from this line of research. And I thought about telling you some master's degree dissertation and some PhD thesis, and telling you a bit about them. Of course, there are much more theses and dissertations in progress, and from these theses and dissertations, several papers and book chapters were produced, okay? But I will talk a bit just to give you an idea. The first dissertation belonging to this line of research was supervised by Professor Motokani. So it referred to the teaching of biology and argumentation. We had also a master's degree dissertation supervised by Professor Fernanda, that was in history of ecology, Another one supervised by me that it dealt with history of evolution, the conceptions on evolution and heredity in the grandfather of Darwin, Erasmus. This other dissertation, supervised by Professor Fernanda, dealt with some ideas from Peirce, from semiotics, and the contributions of Saint-Hilaire, that was a naturalist that visited Brazil. It's not, how can I say, the one that lived at Lamarck's time. It's another one, because at Lamarck's time there were two naturalists, Saint-Hilaire, but this one is a different one. After that, a thesis on explanations in biology about the pollination phenomenon, supervised by Professor Fernanda. We had also this thesis in history of genetics, dealing with the contributions of Walter Sutton to the chromosome hypothesis. This other one dealt with a plan in biodiversity called, I will say, Pós-Graduação of aqui na Universidade de São Paulo. Another thesis in history of biology, in Brazil, the contributions of Sérgio Ferreira to the, how can I say, to the proposal of medicine that deal with pressure, I'd say, and that regulates pressure. I don't know if you have heard about Capitol Hill. So he made contributions that led to this achievement, and he belonged to here at USP, medicine, Faculty of Medicine. This other one about curriculum and pedagogical speech. And in this one, it's about ethno science, dealing with traditional people. It was supervised by Professor Fernanda. This dealt with the proposal of the theory of the equilibrium punctuated by Wood and Eldridge. So, history of evolution. This one deals with popular and folk poetry and biology teaching. Well, now all these works, this dissertation, master's degree dissertations, are concluded and generate several papers and book checks and so on. But now to the PhD thesis, this one is with the path from the modern synthesis to the extended synthesis, if we can consider its existence, and the role of the phenotypic plasticity in that. This one is about teaching and a didactic investigative series dealing with it. this one is about the building of a concept, the concept of crossing over. That is, several students have lots of difficulty in understanding concepts, not only from genetics, but also from evolution and so on. And so if you present how that concept developed, the difficulties which were faced, it can help in the understanding of the concept nowadays, for instance. And so this dealt with history of genetics and also with its application to genetics teaching. This one is another one that shows a bit about some aspects of nature of science, because it discusses the criticisms made by Louis Agassiz to the proposal of Darwin, Charles Darwin. And this shows that new theories usually are not promptly accepted and received. And so this helps to understand the nature of science. This is about ecology and epistemology. This is about, how can I say, scientific technologies and communications in science, about biodiversity, and it is about aesthetics and learning, teaching learning of science. Well, now this is a sample of some works produced in this line, and now supervised by Professor Marcelo, Professor Fernanda, and I. But now I will talk about the research we developed at our laboratory, that is. So we deal with history of biology. We work with history of biology, and in order to do this, in fact, we work with historiography of the history of biology. In order to do that, we need to know about philosophy of biology, epistemology, and also about methodology in history of science research. When, for instance, the methodology, we must choose a theme with a subject, and the students do that. And today it's the object of study, because unless we do so, we get lost. And it's important to choose some kind of research that could bring something new to the area. Even it is a master's degree thesis or dissertation. It is required that we bring something new. We start by bibliographical research in order to know the publications about that subject. Unless you can, how can we say, repeat things that were worked before, the same things and ideas. Since it is a meta-scientific study, because it is a study of a second level, its methodology is not the methodology of science. It's different. In the research sources used in the in this research will depend on the question we asked before. So what you do? You proceed the analysis of original works of the scientists, such as in the original language, including books, papers, notebooks, sometimes correspondence, and also we deal with works from philosophers and historians of science dealing with that subject or its context. In this study, we develop a dialogue between what you see in the primary source and the secondary ones. So you can agree with some historian of science that wrote about that theme, for instance, or not. But you must justify. So this kind of work at first is descriptive, but even so it requires an appropriate terminology, and from where we got this terminology, from the philosophy of science. This part of the research line that we dedicate ourselves aims to study the process of construction, justification, and discussion of biology. So, in addition, this kind of study may clarify why, for instance, as a theory, even if it is well founded, it may be rejected. Of course, the foundation of a theory is important, but sometimes it happens the opposite thing. It's not accepted because there are other factors that can influence that. So we have this kind of works in this line that we talked about, and others. If you use the history of science in the appropriate way, it can help in the study of science and just to understand concepts, theories, models, and so on. As in these works developed at our lab, we studied some historical episodes that showed the difficulties faced by the scientists, the different explanations available for the same phenomenon sometimes, and the non-epistemic factors such as propaganda, struggle for authority in the field, religious convictions that can influence in the building and acceptance of hypotheses or theories, for instance. Is it possible to transpose the historical text, making it accessible to the student? And this kind of text is produced from a historical research may be part of didactic books or textbooks together with the scientific contents, for instance. So, works with development of context and its application to the teaching of biology. Well, I hope that all these research can contribute to the program of Comparative Biology and to the historiography of the history of biology, of science in general, but mainly to a better understanding of the science of today, showing some aspects of its nature, and also contribute to teaching and learning of biology. So, thank you very much. This research, my own research, I didn't talk about it, but I dedicate myself to history, philosophy, and philosophy of genetics and evolution mainly. And nowadays I'm studying a movement which you must have heard about it. That's this new Lamarckism. I studied new Lamarckism in the States. Two paleontologists, Cope and Eliot. And now I am studying in France, Paccard, and other ones. So we thank to this to CNQ, to CAPS, and thank you very much for paying attention during the morning after the last night at Invicta. So that's all.
Questions? Anyone? I get started.
Thank you, Lilian. It was very interesting. I have more as a curiosity. I have other questions that you answered already in your talk. And I was thinking about the last part of the work for the students, the conclusion, the dreadful conclusion. Because it's quite often in our, or mine at least, kind of science, that the students more or less repeat the results on the conclusion. So there is some kind of a barrier that we need to base our conclusions on the data alone. It's not much freedom. And I was wondering in your line of research if the conclusion is more, there is more freedom for the students to express what their personal view on the subject. Was it clear?
Well, what we have in mind is to study, for instance, let's give you an example. For instance, Darwin. So we go to Darwin, and they read, they analyze, and so on. First they go to the original work, and after that they go to the secondary source. Let's deal about the work and, for instance, work from philosophy of science or historical of science. But it's very hard because they must try to go through this contribution considering the science of that time, and try to be the most impartial as possible. What is not easy, because we are, most students are people from the 21st century. And so if you go, it is not easy, and your conclusions must be founded in an analysis very deep, and getting its previous ideas aside. That is what is desirable, but it is not easy to do so. No, we must consider that contribution at that time. No, and so it's not free. If you
Not the personal view.
no, you have to show that. Because there is a kind of problem in writing about history of science, that is the person choose some parts of the work of an author that just show what she or he wants. But this is not acceptable. This must be avoided.
Yeah. Thank you.
I don't know if I answered your question.
Professor, do I have a question? I need that wireless microphone.
I didn't mean to take the stage, but Hilton asked me to come to the front to use the microphone. So thank you for this presentation. I was curious about a very specific point, which is the use of secondary sources for historical research. I thought that was more controversial than you made it sound, as if people would avoid this a lot, but you made it sound like something acceptable as a research topic. And I was wondering, so if you say that historical science is going to be a kind of meta science, so the study of secondary sources is going to be a meta historical investigation, right? It's going to be a history of a history, right? And so would it be acceptable, and do you think it's something that's doable? And to me it sounds very interesting, the idea of contrasting views of contemporary historians, like a thesis that would deal on the views of Ernest Mayr versus Stephen Jay Gould on macroevolution. Would that be acceptable? And do you think it's an interesting topic?
Well, let me answer. Organize my mind. Let's choose, for instance, a subject that Mayr and Osgood had worked with. So, if you go to the primary source that they analyze it, they use it, you must study them, and after that you will dialogue with those historians, agreeing or disagreeing, and showing why. For instance, Mayr, in The Growth of Biological Thought, presents two laws as belonging to Lamarck's theory. In fact, there are four, because he stopped at an intermediate work of Lamarck, that is Philosophie Zoologique, and he didn't see the last versions of Lamarck's work. No, so you can point it out, for instance. Well, and it's necessary to visit secondary source, because these people worked with several subjects, and you must include them, how can I say, giving the reference to their work, because you can arrive at the same, you can repeat something, and it is plagiarism. Yes, if you don't know. For instance, there is an industry of dairy, because there are lots of work about dairy. And so choosing a subject like this, you must bring something new. It's too difficult. And I don't know if there are more things in your question, because it was very...
But it was good.
Is that okay? Always we must go to secondary research with care. No, because all are human, like us. And so one of the works of the historian of science is to revise the previous work. You asked another thing, but now I don't. What we do is historiography of the history of science, because we write about the history of science. But history of science is a meta-scientific studies. In this way, you could consider our work meta-scientific, if you think about it. Thanks. Thank you.
Thank you, Professor Lilian. As Professor Thomas told us yesterday, the line of history, teaching and of teaching of science and philosophy is a differentiator for our program. When we compare the programs that have the same kind of how can I say it? Same kind of weight we have in this campus in the area of biodiversity area. Okay. I could not remember the Portuguese word to perfil. Could you help me? Profile, of course. Sorry guys, too much beers. Okay, now we have 3 international internship talks with our PhD students. Odair, Malu, that's here, Odair's there, and Vinicius is over there. And after these 3 short lectures, we're gonna have a coffee break, okay? So we have 3 now, and I'd like to call Odair, okay? Are you ready?
Yes.
Okay. Thank you, Odair.
Good morning, everyone. My name is Odair Meira. I am a PhD candidate in the Comparative Biology program. I am supervised by Dr. Eduardo Almeida. My supervisor abroad was Dr. Hans Pohl, and the co-authors of this project are Dr. Rolf Beutel and Dr. Brandon Budino. And I'm going to talk a little about this internship that I did in Germany, and the title of my presentation is Believing in New Discoveries, Unlocking the Secrets of Bee Morphology. So giving some context on this international internship that I did, the title of it is Applications of Nano-CT Scan to the Interpretation and Documentation of the Skeletal Musculature Anatomy of Bees. So in other words, is to apply CT or computed tomography to study the muscles of bees. It was developed in the Philetiches Museum in Friedrich Schiller Universität Jena in Germany, and it lasted for 1 year, so 12 months. One curious thing about the Fictitious Museum is that it was in this museum where Ernest Haeckel developed most of his ideas on ontogeny and phylogeny, and also spread Darwin's ideas in Germany. So starting from the very beginning in this project, we had to define what is a bee. So a bee is an arthropod, is an insect, and is a member of one of the most, the big. So, a bee is a member of the Hymenoptera order together with ants and vesps. Talking about the biodiversity of bees, they are divided in 7 families. 5 of them occur in Brazil. There are described in 162 genera and more than 20,000 bee species at the moment. They are present in all continents except Antarctica, and they are vital in several ecosystemic interactions, and they offer and provide several ecosystemic services. So just by this small talk that we had about the biodiversity of bees and the several ecosystemic systems and ecosystemic services that they offer, we understand why it's important to study a bee. But when we go deep into the morphology and the topic that I am interested in, which is muscles, we have that the knowledge on bee skeletal musculature, and this knowledge primarily stems from studies conducted over 50 years ago. So in this overview of the literature in bee skeletal musculature, we have one relatively recent one, which is my master thesis, but the other ones are really not outdated, but that's more than 80 years, and none of them employed CT scan techniques. When we look to the other Hymenoptera taxa, other Hymenoptera groups besides bees, we have lots of these Hymenopteras analyzed with CT scan, especially ants, wasps, and others. And when we deep look at bee research with CT scan, we have only, as far as I know, 3 studies that study muscles with CT scan, and they are very specific. So, for example, three-dimensional atlas of the honeybee neck. The honeybee neck is a very specific region with no more than 4 sclerites and 12 muscles. Functional anatomy of the worker honeybee stinger, again, 5 sclerites and 6 muscles. And this one also that deals only with 1 muscle. When we make the connection of more than 20,000 bee species and only 3 studies that deal with skeletal musculature in bees, we easily conclude that this is a very incipient topic for this group. With this in mind, we delimited our research objectives. The first one and the main one was to expand the knowledge on the anatomy of bees using CT scan technologies, and the specific ones were to describe internal and external morphology of the skeleton and the muscles. Another one that we had as an objective was to update anatomical concepts for the group and to test homology hypotheses proposed in the literature. Before going to the methodology, I would like to make a mental exercise. So let's pretend that we have a bee in front of us and we make a sagittal section in it. What would we expect to see would be the brain, the hemolymph, the cells, and also, or specifically, the muscles, which are here highlighted in reddish, and everything that's not reddish is the skeleton of a bee. The association between the muscles and the skeleton is what we call the skeletal musculature association. So, in other words, the objective of this study was to understand this association. For this, we selected 2 species and 1 specimen from each one of these species. The main star of this study is Tarmius albomaculatus, and for a comparative step, so for things that were really different in Tarmius regarding the previous literature, we compared to Alasioglossum, which is a different family of bees, and basically both of these specimens, they underwent through the same protocol. I'll not go into further details in terms of the CT scan techniques, but I can answer this after the presentation. But they were analyzed in KIT, Karlsruhe Institute for Technology. It's a synchrotron facility. All this raw data was, after that, prepared and analyzed in a mirror for the segmentation step. And everything that was segmented there was then imported to GStudio for the volume rendering step. The first result of this study was the delimitation study and the construction of every single sclerite. So every single small plate that you see in the knee was reconstructed as a single material. So now we have the complete morphology, the connections, and of course, as this is the main topic of this research, every one of the 205 muscle groups of the bee was reconstructed also individually in terms of origin, insertion, if it is or not ramified, and the interaction with the skeletal musculature. And now, bear with me because I'm going to explain each one of these muscles to you. Now, I'm going to highlight just some of the main findings of this study. Everything for me is main because I spent like more than 8 months doing this, but I'm going to highlight some things that you might relate to. So the first thing that we did was we present alternative interpretations on bee anatomy. So we have a lot of concepts in bees that have some discrepancy between authors and between the same authors. So this is a table that highlights the application of the terms for the labial sclerites in bees. And we can see that Snodgrass 1925 disagrees with Snodgrass 1942 in terms of the terminology for this sclerite. And we have that Snodgrass 1942 disagrees with Michener 1944. So we reconstruct every single of these sclerites and we compare with other bees, with other hymenopterans, and with other neopteran insects to provide a pattern, a terminological clarification for this term. So in this case, our approach in this structure was to divide the labial into the prementum and the postmentum to be subdivided as a lorium and a median lobe of the postmentum. This was the same approach that we used for the complex of the endoskeleton of the head. It's a very three-folded complex structure, and we compare to other bees, to other hymenoptera, and to other insects to understand all the fusion of the tentorium, hypostoma, postgenital, the membranes, the breeds that they fuse together. We applied the same approach for the lateral sclerites of the throwers, where we compare to other bees and to previous literature, and to other hymenoptera that also have data with statistical technologies. And we did the same also for the sclerites of the skin, and also for the musculature that was not new but already described in the literature. Another important result of this study is that we documented new skeletal musculature groups that were not present in the literature, so it was the first time that we have this. So on your left, we have the skeletal musculature, or the muscles of the mandible in the previous literature, so it was divided in number 9 and number 8, so a ductor and a ductor, one muscle that opens the mandible and one that closes. And when we were doing the reconstructions, we discovered one new muscle, so a third muscle of the mandible. You can see it here, OMD6. It's a very small muscle, so it's pretty understandable why it was missing in morphological studies. And we have a hypothesis for this muscle that it don't act like an opener, or it can't move the mandible because it's so small, more or less 3 or 4 fibers. And we believe now that this is actually a proprioceptor, so it more or less sends information to the brain if the mandible is under pressure, instead of moving the mandible. Another good result that we had in this study was the discovery of more two muscles in the abdomen or metasoma of a bee. They are, again, very small and hidden between the segments, and together with 3 muscles that are already described, they compose the 5 lateral muscles in bees. This is the first time that we have this result, but when we go to Snodgrass 1956, in this figure, we have that For the basic insect or a grasshopper, we have 5 lateral muscles in the metasoma or the abdomen, and this is the same pattern that we find in bees. And also, if we look to the connections of these muscles, 3 of them attach to the apodeme, and this is also the same case that we have in bees. Am I saying that they are homologous? No, but for the one who really loves like morphology, it's quite interesting to have these 2 new muscles that were never been described, and we have the same pattern with the dorsal. Another thing that I was able to do was to revisit my master thesis, and in this paper from 21, one of the main findings that we had was to describe a specific topology for the musculature of the tubular maxillary in a family of bees, the Halicteres. And the important thing is that we have 2 complexes, so a labial and a maxillary. And when I was doing the comparative step with Lasioglossum, which is a bee from the Halictid family, I found the same pattern, so the same 2 complexes, a labial and also a maxillary. And we can see how different it is from the Tyreus albumaculatus. But the fun thing is that I see that I was not crazy when I was doing the sections, because it was really hard, and apparently I was at least somehow correct in this point. The conclusions and future directions from this study is that now we have the first 3D atlas for bees, not only with muscles, skeletal musculature, sclerites, everything together in a complex. We present new interpretations of bee anatomy. I just highlighted one of it, but we have several new interpretations for the terminology. We presented standardization that allows the comparison with other hymenoptera, such as ants and wasps, that are also studied with CT scans. And we believe that we present with this project, we have new possibilities for morphofunctional research and also phenomic research. These are the references that I use in this presentation. I would like to thank my funding agents, FAPESP, Friedrich Universität in Vienna in Germany, Universidade de São Paulo, my university, LBCA, my lab here in Brazil, for all the support, and of course Entomology Group in Vienna for receiving me, and all of you for your kind attention. Thank you.
We have time for a few questions.
Just like a curiosity, so you said this is not very specific, and I was wondering if in these studies of muscle anatomy in insects, there are studies that analyze like development, if there are people studying like using development to try to understand like muscle homology, and if this is a perspective that people have like in entomology, because in vertebrates I know that is people looking through development is a very useful way to try to understand homology and muscle divisions and various traits. So I was wondering if with insects people also have this perspective when there are studies like studying development to try to understand this diversity in muscle anatomy.
Yeah, they do. I'm not one of these persons. I'm more like a structural morphologist. And when I was discussing that small muscle that I was talking, like taking the mandible, we went through the same discussion. Like we have OMD6 or OMD8 as possible candidates for this muscle, and we were discussing, okay, so the position is in the inner ring of the mandible. So by the topology, we believe that it's OMD6, but to be fair, we'll never know unless we do like development or maybe genetic analysis. But for structures, I was just like looking to compare with outgroups and seeing topology. But there are people studying development for the homology of muscle, of course.
Just a quick addition is that it's good to also remember this is about the metabolism insect, so it's going to change immediately when it becomes an adult. So some parts like the legs, they don't exist as a larva. So it's something that arises during the very last part of its post-embryonic development.
So also
get all the muscles in the pupa stage, so it's in the very last part of the life. But people do it, as Adolai said, people do look at this last part. It's also informative. It's still important for understanding this.
I have a curiosity, personal curiosity. We have a postgraduate program in entomology in our department, but you choose comparative biology. Why?
Yeah, I guess I answered this once or twice, I don't remember. But it's complicated because I was thinking to apply to comparative biology or to entomology. I had like that idea because I'm a master in entomology. I wanted to do like both, to have like entomology and comparative biology instead of entomology and entomology. And also, when I was like applying and talking to my supervisor, I think comparative biology opened the process first, so. But the main reason is that I wanted to have both entomology and comparative biology in my study.
To characterize the muscle associated to the mandible as a proprioceptive muscle, did you look for, or are you going to look for a receptor? Because the only receptor will tell you it's
the nerval.
You can find proprioceptive receptors within muscle tissue, so which measures the degree of contraction.
Mm-hmm.
So, say it's a proprioceptive muscle, you have to look for a receptor.
Yeah, it's because when you look to the axis of the mandible, even if it was like a strong muscle, I think it will be not like that efficient, and it's a very small muscle. And this one is actually big, but if you look to lasoglossum, it's almost like 2 fibers, and it goes to this ring, and we see like the nerval coming from the brain, and it attaches. So it was like a discussion. It's very incipient, the discussion. We're just like.
The nerve could be just the innervation of the muscle,
Yeah.
but transmission electron microscopy will answer your question.
Mm. But you think that it might be the case that when the mandible is under pressure or in tension.
What
it
do
might
I
say
understand
something.
about bees?
But you understand about muscle.
Yeah.
But I think like when it's under tension, I don't know.
No, I have to see the mandible.
Yeah. Okay.
Sorry.
We can discuss that. Really good.
Thank you very much, Alô. Next one will be Malora Almeida with her African catfish at Philadelphia.
Good morning, and I am Malu Almeida. I'm a PhD student here, and my supervisor is Professor Flávio Bockman. I'm here today to talk to you about my time at the Academy of Natural Science in Philadelphia, and during my internship. And my supervisor was Mark Sabay. And first of all, I would like to say thank you to be here today, watching my presentation, and also thank you for the organizers of this meeting for inviting me. And just to let you know, my presentation has 2 main parts: one about what I study and what I was in Philadelphia studying African catfishes. And the second one is more about personal experiences. And let's start with catfishes. So I study catfishes, and I don't know if you know what a catfish looks like, but if you know, you must be imagining something like that, because they are common as a food source, or because they are very popular among aquarists, like this guy here. But catfishes are very, very different. So the order has 40 families, and if you are talking about fish, this means a lot. They are worldwide distributed, and we have some groups occurring in the specific areas like North America, Africa, and we have entire clades occurring in specific areas like South America and Asia too. There are so many ways to study all this morphological diversity, but the one that I chose was looking to the lateral line canals. And what is the lateral line system? It's a mechanosensory system present in fishes and aquatic amphibians, and the system is very important in different behaviors such as schooling, finding food, avoiding predators, and the functional units of these systems are the neuromass, and we can find two types of neuromass. But what I am looking is the canal neuromass, and specifically about the patterns of the canals in the head and the trunk of the fishes. And additionally, the canals are associated with some bones, and we have a very preserved canal-bone association. So if we can look to the canals and understand the patterns, we can also understand some school modifications in fishes. And of course, the canals have been used like a taxonomic tool in different groups, and the lateral line canals are also have been studied like in a phylogenetic approach. And this paper here is from a former student from this program, and with a very popular group of fishes, which have the tetras and piranhas. And we don't have a study like this to catfishes, but I hope very soon we can have one. So, of course, if you have a worldwide distributed group, you have some gaps. So we don't know much about lateral line, the patterns of the lateral line canals in catfishes from Africa, from Asia, and that was I was trying to do in my internship. I was trying to study the lateral line canals in the Big African clade, which has all these families here, and also this one, but this one is not from Africa, it's from South America. And to study the canals in this group here, I have to find a place, an institution, with these specimens. And that was the reason I was in Philadelphia. But first, I just want to keep this in our mind, this critical point of view, because we are going to reach countries to study specimens from different parts of the world, because some economic and historical reasons, so we need to remember that. But let's go back to the positive vision of the world. So I was in the Academy of Natural Science of Drexel University, or just the Academy, and I don't know if this institution is familiar to you, but it's the oldest natural history institution of America. And when we have an institution old like this, with such long history, we is expected to find some interesting histories about people that passed through that, like Ruth Patrick, this lovely woman, and her legacy. And I'm really like about what she did, and it's good to see that her legacy is still alive in the Patrick Center and the Academy. And of course, the real James Bond, an ornithology that who inspired the character. But I was there, of course, because of the ichthyology collection, which is taxonomically diverse, especially for catfishes. and with geography scope worldwide. So it was everything I was looking for. And I can talk to you more about this collection and how it is a special place, but I don't have time for that. And just to show you something that I think is very special is this tuna here sent by Hemingway. And a collection like that is a product of years and years of work. And we have some important names as a curator of this collection, but I'm really grateful for these guys here, of course, because of all this, but especially these three guys here, which are currently working at the Academy, because they improved the catfish collection. And it was in that scenario that I could analyze different preparations of all my in-group, at least one specimen from my in-group, and also at least one specimen from my out-group. And I could apply a standardized, I hate this word, I'm sorry, standardized canals nomenclature and terminology for the parts of the canals, and we can compare the data. And some of the results of my project during my internship, we were able to describe the canals of the Big Africa clay, and for some of these guys here, oh sorry, it's not working so well, I don't know why, okay. So for some of these guys here was the first time that we have data about lateral line canals, and I'm going to pass very fast through this part because it's kind of boring to you, but we have some difference, some features in specific parts of the lateral line canals in Big Africa clay. Some of them are very interesting when we are looking to how these conditions are distributed in a phylogenetic regarding phylogenetic relationships. And we have some parts of the canals that may be present in some groups or not. So, and we have some characters about the morphology of the tubes and pores and how they are distributed in the phylogeny. And about some canal bone association with some bones present, the presence of the scapula. So we have two families that we don't have this bone associated with the temporal canals. And all these results are being incorporated to my PhD analysis, which includes all the families. But I just have a PhD now, a dissertation, because my time at the academy and all the material there, and I'm so grateful because we have this kind of institutions and we need to take care of them. Okay, so I'm not too old, but I'm 30 now, but I think a few things in life changed us as living in a different country during internship. And some days could be really awful, and no one is giving you a survive guide. But if I can tell you something, what I would like to share with you, let's start with some things that we need to be alert to. If you are going to spend the winter, stay warm and never turn off the heating system, even if the owner of the house is telling you to do that. Don't do that. You can get in trouble. And bureaucracies, they do not start when you are leaving the country, at least 6 months before, and they do not end when you return. So CAPS will knock your door and, hey, you need to send me some documents at least 6 months after you return. And of course, the challenges are about talking a different language, but something that I learned was you are the person doing the hard work there. You are the person trying to speak their language. So be proud of yourself and go ahead. And what about what was positive in my experience? I was living in a good and safe area, and that was really good. And a full language immersion, so I wasn't talking Portuguese during my internship because I was surrounded by people talking English all the time. That was good, actually. I could improve my English. It's not so good. Yeah, it's better. A full access to the specimens and in the collection, and also the equipment in the lab was really good. I could develop my project very well. I wasn't halfway to by bus or train to two big collections, and I could use both in the Theology Department again, and I'm really thankful for that. And I had a good supervisor, and the environment and the people in the academy was they are so kind, and I was really happy to be working there. Of course, you're gonna find your way. Just try to enjoy your time. Time flies, and I'm really happy with my memories and all the people I met. And I really miss some of them, especially these two girls right here, Dust and Basil. And thank you again. Thank you, CAPS, for the financial support and all the institutions, the Comparative Biology program, my lab, and all the institutions I could visit to analyze specimens. The academy staff, and especially my advisor, my supervisor, this guy right there, Mark, who was also my host. So thank you. They are really, really special and amazing people. My advisor Flavio and Pedro and Murilo, because they shared with me their passion about morphology and the electrolyte system, and I think that's all. Oh, I'm sorry. Thank you again. I'm just kidding. You can ask me this good question, but I will answer in Portuguese.
What's the meaning of life?
Sorry.
A difficult question. What's the meaning of life?
I don't know. I have no idea.
42.
42, yeah. You're right. Few questions, guys?
How do you study the channels? Do you like cycle?
No. This is no. I'm using clear and stained and double stained material, but I'm also combining some different preparations like muscle preparations and CT scans, dry skeletons, and also ethanol preserved specimens. So I'm combining different preparations. But one important thing is we need to keep the skin, because we have some part of the canals going into the skin, so we need to keep the skin. I'm not dissecting because of that. By the way, congratulations. Your images are,
Go morphology.
yeah, morphology rocks.
Thank you for the lecture. Good to see morphology in the top of their study.
Yeah.
And what do you think about the molecular part of the biology when you went to a foreign country like the United States? Because when I talk to colleagues from Europe, for example, they are all the time dismissing the morphology in their studies in plants. I don't know if it's the same case in animals too.
Yeah, if you are working with phylogenetic relationships, just looking for, trying to understand some groups, and people will ask you about what you are not doing molecular studies. But at least in my experience at the academy, doing just a morphological study is something normal, and people are okay with that. We have so many things to understand regarding morphology, and I don't know, I know molecular studies are important and we need them, but we also have a lot of work to do here. So I think, yeah, it's a lot of pressure in US to work with molecular data, but I don't know if it's something like, yeah, so
Not so pressure.
no, the academy we are, I was really comfortable doing my work.
Questions? More questions? Thank you very much. Now we have Vinicius and Ellie with the last one in this part before the coffee break. And he's going to present shapes of lizards in America. Isn't intentional America? Of course.
Okay, so I'm going to try to speak without the microphone because it's really loud and I move a lot, so might happen something here. So yeah, as Milton said, my name is Vinicius. I'm a PhD student at Tiana's lab. And I'm going to tell you, this is cool because I didn't know if that was intentional, but I'm also going to talk about morphology, but through a different perspective. So it's the third talk in this session, and it's also about morphology. But here I'm going to talk about my PhD work with morphological diversity in lizards. Let's see. Okay. So this is a lizard. Another lizard. Another species of lizard. This is also a lizard. This is a cool lizard that shoots blood through his eyes as a defense mechanism. This is also a lizard. This is also a lizard. And this is also, those are also lizard, although Wilfred is here, he might disagree. But let's consider that those Osminians are lizards. All of those lizards, they are in the American continent. So you have some of those species in South America. So for example, this is the species that Danilo talked about yesterday. You can find it here. Also, this species is very common in our campus of Urbana. But also, we can see that there is a distribution of those species in the continent. So one of the ways of studying morphology is not just to characterize its variety. So we can see different shapes and ways of looking like a lizard, but also to look into its distribution. So distribution studies, they are mostly focused on the distribution of species richness. So which areas of the continent or of the biome or of the world, they have more species or less species. And for tetrapods, the pattern is pretty much similar. So we have a tendency, it's not very clear, but those lines are the equator. So we have a tendency of having more species richness around the equator. And the explanations for those are many. I'm not going to delve into that right now. And we also have, talking here more specifically about lizards, other studies, but those focusing on specific groups that are looking to different aspects of diversity. So for example, phylogenetic diversity or functional diversity. But it's really hard to see studies, especially with vertebrates, that look into the distribution of morphological diversity, because that's something that's not that easy to access. You have to go to a collection, or you have to collect this material, and you have to measure it with your own hands. So this is the gap that I tried to fill during my PhD. So my first main question is if the distribution of morphological diversity is congruent with the taxonomic diversity in lizards. So do we have more diversity of shapes of lizards around the equator, or are those patterns different? And my hypothesis is that they are different. And my second question is if some environmental variables such as temperature or precipitation, they are correlated with the distribution of this diversity. And I'm going to talk also about this further. So yeah, that leads me to why I went to New York. So I spent 1 year in New York doing a BAP fellowship internship abroad. I was at Anna Carnaval's lab at City University of New York. Her lab is focused on biogeography and evolution and ecology. So she works with mapping diversity and understanding diversity through many perspectives. And for me, that was really cool. That's what brought me to New York to work with her. She's from Brazil, she's from Rio, but she's a professor there for over 15 years now. But also the second reason I went there is because in New York you have one of the largest museums in the world. So don't get me wrong, we also have amazing museums here in Brazil. But as I was interested in the global patterns, here in Brazil you can find a lot of species, at least for lizards, from South America mainly, but also a bit from Latin and North America. But if you want to access species from all over the world, you have to go maybe to US or Europe. I'm not going to talk about colonization here, I don't think we all know that, but you can find those things here. And then at the American Museum of Natural History in New York, you have one of the 5 largest herpetological collections in the world. And they say that they have specimens from Africa, Australia, Madagascar, New Guinea, the Pacific Islands, Panama, South America, and so on. This is one of the most iconic collections. The city itself is very iconic. You've seen it in movies. That's a picture of me with a Komodo dragon, which was really cool for me. This is an adult Komodo dragon. And this is the famous T-Rex that is in the exhibition there. And that inspired the Jurassic Park movie that brought a lot of people to biology, at least from the 90s. So how do I access the morphological diversity? So I go to those collections, I get those lizards that are preserved in alcohol, and I start to measure them with my caliper. This is just to show, you don't have to see which measurements I took, but this is just to show that I took measurements from head, trunk, wings, and tail. So I try to measure everything from a lizard as far as I could. And also I didn't only use specimens from the American Museum of Natural History, but also USP here in São Paulo, our collection here in Ribeirão, and also other collections from Brazil and Europe. So just a basic overview of the framework, so the analysis that I did. So basically here, what I think is, I put together geographic information. So I look into the distribution of each species that I access. I combine that with phylogenetic information. So we have good phylogenies published in literature for squamates, and I can use that to take into account the relationship between those lineages. And I also consider the morphometrics. In my case, it's not geometric morphometric, but linear morphometrics, because I'm using a caliper to take linear measurements. I also get environmental variables from GIS, from remote sense base, from satellite images, so I can get temperature, precipitation, topographic information, and so on. And I put all of this together in R to see how those distribution patterns they're going to show in the map. So as I'm going to show a lot of maps from now on, this is a map of Middle Earth, for those of you who know Lord of the Rings. So I put this map in a grid so we can see those small squares. And we can look into each square to ask ourselves some questions. So for example, if you go to Rivendell, the elf land, which species and peoples can you find there? So if you look into this square, this is Rivendell, you can find this group of species and this group of things. But if you go to Mordor, down south, in the southern region, you're going to find different types of species. So this is something that we also do in biology. We can look into different regions of the map. We can use small squares and small grids to see and compare diversity between different localities. This is the way we do it. So okay, now talking about my sample. So I got to sample over 5,000 lizard specimens, which represents almost 1,500 species sample. That's pretty much a fifth of all the diversity of lizards described so far, living lizards. And in terms of genera, I sample almost 400 genera, which represents 65% of all genera described for lizards. So that's a very good sampling in terms of global diversity. I have all families of lizards described, including this study. And for me, it was pretty special because I really like to get this diversity with my bare hands and touch it and see it and think about it and measure it. So this is a map of the taxonomic diversity that I sample. This is not something new. This is just this is something that has been done to look into the distribution of species regions for squamates and lizards. But the cool thing here is that we can see that the pattern that you see in other studies is similar here. So we have a considerable diversity around Amazon region in Central South America, here in Africa, but also the Indo-Malaysian range, and also Australia. So this is following pretty much everything that came before. But this is for taxonomic diversity. However, there's a lot to be said about this diversity, and for today I'm going to focus on one part of this map, which is America. So I went to the US, and in the US, if you say America or America, they're going to think we're talking about themselves, because they only talk about themselves. But I'm not talking about their America, I'm talking about the real America, so I'm talking about the American continent. So I just wanted to make this joke, and we're going to talk now about the diversity patterns in the American continent. But yeah, for my PhD I'm going to discuss other things, but I can include that now. So this is the taxonomic diversity in America, on your right, or actually on your left, and this is the phylogenetic diversity, just to see that this is pretty much similar. So you have a distribution of those clades that seems to be concentrated around the equator, in this region here, and those two things they follow. And as I say, this is nothing new because this has been done before, but this is new. So here I got all the diversity that I measured, and I put that in a map. And if you look into these maps, colors that look, or we like to say hot colors, so colors that are more red or orange, they represent a higher level of diversity. And we can see, for example, let's look into some regions, that in the Amazon region, we have a high diversity of species regions, but that doesn't follow when we're looking to the morphological diversity. But we do have a higher diversity of higher morphological diversity in the southern part of South America, but that is not following species regions, and we can also see in North America those patterns that are not congruent. So we can see in those maps that species richness and morphological diversity, they don't seem to be congruent, but actually tested that. So I use a correlation test that takes into account the spatial correlation, and actually found that there is statistical significance to say that there is no correlation between taxonomic diversity and morphological diversity. So this is my first question that was answered for America. But I also test some environmental variables. So I don't know if you can see here, but here we have mean temperature, diurnal range temperatures, temperature seasonality, precipitation, an aridity index, topographic ruggedness, and habitat heterogeneity. So for taxonomical diversity, the main environmental variable that explains this distribution seems to be mean temperature. This is really cool because this follows the generalized patterns of distribution of diversity, but this is for species richness. When we go to morphological diversity, the variable that is more correlated with these distribution patterns is actually habitat heterogeneity. So habitat heterogeneity, for those of you who don't know, this is a map of the habitat heterogeneity in the American continent. So the idea is that if you get, you guys can see here, but if you get a grid cell, I'm going to measure in this grid cell, for example, we have cane sugar here, we're talking about Ribeirão, and then we have here a forest like Mata Atlântica, and here a bit of Cerrado. So this would be a dissimilarity level of 3. But if you get another grid level that there is only Cerrado, this would be a similarity level of 1. So the habitat heterogeneity here is higher than here. Does that make sense? Yeah. So we have a higher availability. Yeah, thanks. We have a higher availability. availability of different environments and niches here. And that makes a lot of sense if you think about morphological diversification. Some of the theories say that you will have more species looking different in regions that have different niches available. Okay, so for the main questions that I tried to answer, for the first one thing is not congruent with the other, and the second one is that higher morphological disparity is associated with higher habitat heterogeneity. But we are never satisfied, so I look deeper into that. So one thing is that one of the components here is that I'm looking now for regions that contribute more to this morphological diversity. And we can see that those regions here and those regions here, they seem to have a very high level of morphological diversity when you compare to the diversity overall in the continent. And if you look into a biome map, you guys are going to notice that this happens in Caatinga, Cerrado, the Atlantic forest, especially in the Mantiqueira region, so southern Atlantic forest, also in the Chacos, and there's also a bit here in the eastern temperate forest around the eastern US. So those regions seem to be the more important regions in terms of the morphological diversity. So this is really cool because we can start to think about what do these biomes have that favor the rising or the permanence of these high levels of morphological disparity. Another thing is that I got to measure turnover. So here on your left, you have the taxonomic turnover. So what the turnover is showing here is that regions with colors pink, orange, or yellow, there are regions in which you see a change of species composition. So you guys can follow, if you remember the topography of South America, this is pretty much the Andes. Here you have some high elevated regions. So you can see that those regions, they represent an important part of species composition change. And when I tested for environmental variables that I mentioned before, topographic ruggedness, which is the measurement that I got from topography here, seems to be correlated with those patterns. But when I mapped the morphological disparity turnover, this changes. So you can see that the patterns, they don't feel much the same. I don't know if you guys can see, but here you have like more blue in these regions than here. Here seems that the composition of traits doesn't change that much. And also when I test that, I found again that habitat heterogeneity seems to be an important environmental variable to explain these patterns. So this is also really cool because we're starting to look into how these patterns of diversity are changed in a spatial scale. And also here I got to map the traits. So this, I don't have much time to explain a lot of this, but the idea here is that you guys can see some changes in color, especially here and here. So we noticed that regions around the tropical of Capricorn, that's the one that goes here in São Paulo, you have a tendency of assemblages to have species with smaller limbs, longer bodies, and also smaller heads. This is really cool because among lizards, as I showed before, we have species that are limbless, elongated, and that are fossorial. So they use fossorial contexts as their main habitat. This is a very important component of the diversity here, and when we plot this morphological variation in a morphospace, we can actually see a very clear distinction between here are all the species of my sample that look like a lizard. Here are species that look more like a snake but still have like smaller limbs and are elongated. And here are species that are very elongated and completely limbless. And this is interesting because when you think about their habitats, here are species that are mainly arboreal and terrestrial. Here are species that are already fossorial for leaf litter or sand swimmers. And here are species that you can find like 3 meters deep in the ground buried. So those are true borers. So this seems to be a very important component of that diversity that I show in the map. And this also goes to what I'm now looking into for this part of my PhD, which is the idea that how can I look into that thinking about climate change. So for amphibians, for example, we already have studies showing that amphibians tend to have more fossorial species around the 2 tropics. So this seems to be a pattern followed by at least species of lizards in the American continent. And this is really cool because when you think about climate change, we can think about fusariality as a sort of refuge of environmental variability. So we tend to have fusarial species in environments that are more harsh, so environments with lower precipitation, less complex vegetation. This is also association with a lower species regions, and also regions that if you look into historically, they have more unstable climates. So climates that throughout the history they change faster than other parts of the world. So this is something that just some ideas because I'm looking to that more now. But I think that for now this is it. In terms of a broad internship, I think apart from being able to access material that I couldn't access here, going through a collection, I think that a lot of these perspectives of this project, it changed a lot when I went to another lab and when I learned to think about my own project through different perspectives. But also going abroad shows me one thing that I think sometimes we don't appreciate here, is that we do amazing science here in Brazil, and people abroad, they know that and they value that, they really love. I think that Malu and Odair, they agree. They really love Brazilian researchers because they know that we are good. So I'm really proud of the science we do here too. And with that, my acknowledgements and thanks for your attention. If you have any questions.
Questions?
First of all, thank you for your lecture, and I love the Tolkien's reference in that slide. Okay. Well, I have so many questions, but one of them is, for example, the first assessment that you have, morphological and taxonomic diversity are not always covered. Okay. But in your opinion, when you have a researcher that works with taxonomy, for example, they have a kind of a view what the species are,
Yeah.
but it can be not so clear. For example, in what grade do you believe it affects the results in studies like that, for example? Because you work with names of species,
Yeah.
okay? And taxonomists name the species.
Yeah. Yeah, this is interesting because, so yeah, we can ignore that species. When I say that there is a lower species morphological diversity, that doesn't mean that everything there looks the same. But if you put that in this perspective, so when you think the pattern overall, you're going to start to see that there might be a bigger or not variation. So for example, there's one group of lizards in the Andes, which are the neolemos. They're pretty much like our columbos here, and they are very diversified. You have a lot of species distributed in different altitudes and everything. But when they put them here, they're all in the same area. They all sort of look like the same. So I think it's a matter of perspective. I try to get traits here that I can say that they are associated with functional aspects. So how do these animals behave in the environment? And I think that if I look into groups more specifically, so if I just look into Lialinus, I might find different patterns. So I think that depends on the taxonomic level that I'm including in my study. I don't know if that answers your question.
It's surprising that, because so in some cases you didn't work it with species names, but with groups of functional species.
No, I
Is that
work
right?
with species names,
Uh-huh.
but one thing is that when I was collecting this data, I was just seeing what I had available and then getting everything that I had available at collection and measurement. So I didn't look into how this lizard looked like or anything like that. I was just measuring it to see, because I didn't want to bias the morphological diversity that I was sampling. But yeah, I think that now that we have, at least for squamates and for most tetrapods, phylogenies that combine morphological and molecular information, it's really good to follow the phylogeny to decide what you're going to sample, at least if you're looking to the phenotypic diversity, because then you're not biasing your samples just thinking about shapes, for example.
Thank you.
So I have a question, and it may look kind of basic, but you measured a lot of specimens from America, and we know that there are knowledge gaps regarding the geographic sampling for any group. And you retrieved some very, very good maps showing the morphological diversity in very specific places and very small squares. My question is, how can you overcome knowledge gaps, geographic gaps, and how did you retrieve those maps? There are kind of extrapolations or some kind of analysis that makes inferences based on year-by square or something like that.
Yeah.
The question is, how good your sample needs to be in order to make those inferences?
This is a great question, actually. So I think this is one of the challenges of working with a database that is this taxonomically diverse, is that I'm not an specialist in everything here. So we have a pretty good published database of distribution of lizard species. That this is different from GBIF or IUCN, because this was made by specialists, and you have some researchers that are reptile specialists that are always curating this information. So I trusted that. But one thing that I could do is to get this information and try to treat that a bit more. So I could check for every species that I'm including all the literature published and see if those points make sense. This could be something that I could do. But I don't know if for this size of sample that would make much sense. As this is a database that was published by specialists, I tend to trust it. But I also think that when you have a pattern that is this big, some arrows like saying that the species goes until Saint-Simon, but it's actually saying the polygon that goes until Ribeirão, that might make a difference if you're looking just into the São Paulo state. But if you're looking at the whole American continent, that might not be much of a difference. So this is one of the things. The other thing is that one thing that I can do here is that I choose metrics when I mapped them that consider the species richness that I'm including in my study. So I could also, and this is something I probably do, there are ways of getting this and simulating a distribution that would be random and then comparing these two things. This is something that people have been doing a lot, not just for maps, but also for comparative analysis, is that you sort of produce a model random pattern and you try to compare how much the pattern you found is different from that. And that goes through like 1,000, 10,000 simulations. So you see like there's a chance that what I'm seeing here is not a bias or is not something that would fall into expectation. So there are statistical ways of dealing with that. But for example, there are some issues we could mention. So for, if you think about taxonomic diversity, I would say that this is one of the this is one of the regions in the world that has less number of species that we should know that they are there, but we don't know. But when I see this sort of pattern, so this is the opposite of what you expect, right? Because if this is undersampled, I would expect here to have a lower richness. So those kinds of things, when I'm interpreting my results, I have to take into account. If I had a map that shows that here is the higher number of species, I could say, well, but we have São Paulo here, which is one of the leading regions in Brazil in terms of collecting and having access to species, at least for lizards and I think that for most groups. So I have to take that into account. But when I was presenting that to our lab a few weeks ago, I was mentioning that, for example, I think that Lilo asked, why is red here? What does that mean? For me, it doesn't mean a lot because I'm not looking to that specific pattern. So that can be a bias, that can be something very cool. But I'm actually looking to the map as a whole. So for me, it's more about the species pool and the large assemblages than specific things. So I think that depends. At Anna's lab, they work a lot with the Atlantic forest, so they go to very specific localities. So for them, a very thorough sampling, that might be something that makes a lot of sense. And it's also something that can be done in due time.
Thank you.
Yeah.
Vinicius,
Yeah.
since you measured species from around the world, monitor this, so are you going to test your hypothesis in other Africa, Asia, Australia, just to confirm your findings from the Americas?
Yeah, I will do that. It's just that I have a lot of data, and I have many ways of dealing with this data. So, for example, this whole thing about the fossorial species, this is something that I also start to see in Africa, because in Africa you tend to have a lot of fossorial species around the tropics also. So, this is something that I will do. I will look into the global patterns, and again, when I go to the global patterns, I can look, for example, into the biomes, or I could, but that would be also a bias. But yeah, that's something that I'm going to do. And I have other things that I want to do here, so yeah.
Okay. Basis. In plants we have a joke, because do you know what part of São Paulo state is the most diverse in terms of plants?
In terms of plants?
Yes.
Would be around the Serra do Mar? No.
Along the Anhanguera Road.
Right.
Because the
that's
collectors,
where,
yes.
yeah, that makes a lot of sense. That's one thing, when I saw this pattern here, so this is New York, right? And okay, I'm in New York and this is a pattern that shows, but when I see into the species richness, this is actually another region where I got more richness of species, so I can compare these two things. But yeah, that also makes a lot of sense.
Okay. Thank you very much.
Thank you.